Hello, I'm Nelson Freimer, professor of psychiatry at UCLA and director of UCLA's Depression Grand Challenge. On behalf of the organizing committee, I'd like to welcome everyone to this symposium entitled Human Neuropsychiatric Disorder Research, From Bench to Population. In approving Proposition 14 last fall, Californians voted to invest in research that could lead to transformative advances in our ability to prevent and treat mental illnesses. Through the scientific presentations in today's symposium, we aim to convey both the exciting neuroscience research opportunities that Proposition 14 can enable, but also the challenges that we must address to ensure that our discoveries decrease health disparities rather than make them grow wider, that their benefits are available to all of our people. And now for an additional welcome, I'm pleased to introduce UCLA Chancellor Jeanne.
Thank you, Nelson, and good afternoon, everyone. And thank you for joining us for the symposium. So research into human genetics and stem cells can play a major role in helping us understand and treat neuropsychiatric disorders that affect brain function, emotion, and mood. Investments in this research are critical, especially now as people around the world continue to grapple with the mental and emotional effects of this pandemic. The mental health toll over the last 12 months has really been extraordinary. We've seen a percentage of adults in the US reporting symptoms of anxiety and depression increase from 11% in the first half of 2019 to 41% in January of 2021, an astonishing increase. Notably, this has been felt unevenly by different demographic groups. The passage of Proposition 14 in California came at an important time. With $1.5 billion for stem cell research earmarked for neuroscience research, Prop 14 has the ability to create remarkable opportunities to transform our understanding and our ability to treat mental disorders, to support the tremendous scientific expertise of California institutions and drive wide-scale collaborations, and to ensure that these advances benefit the diverse population of California and beyond. The work of many of our partners here today has already brought this state to the forefront of stem cell research and laid the groundwork for cures for neuropsychiatric disorders. As a public university, UCLA has the duty to work with our peers to pursue these kinds of advances. Finally, I want to express my deep appreciation to our hosts, speakers, and guests, and to the Bloomfield Family Foundation, whose generous endowment makes this symposium possible. Thank you again for taking on these topics and continuing to advance this work for the benefit of our state and the nation. I hope you have a productive afternoon. Thank you.
Thank you, Chancellor Block. I'd now like to introduce Garen Staglin. From his perspective as chairman and founder of One Mind, co-chair and founder of the Healthy Brains Global Initiative, and as a tireless advocate for science and support of brain health, he will set the stage for the scientific presentations that will follow. Garen.
Nelson, thank you so much. It's such a pleasure to be with all of you, and I'm honored to be with such an illustrious group of scientists and participants. As Jeanne said, and as you said, this Proposition 14 is a watershed for the world and global neuroscience community. And California, as the 5th largest economy in the world, is setting the stage for what we hope will be a dramatic acceleration of research. Before I give my few comments, it's my great pleasure to introduce Bob Klein. For those of you in the CERM family, you need no introduction to Bob Klein. But if you're not, Bob is a tireless advocate to eliminate chronic illness. He has, as I, a personal reason to be engaged and involved in, and I've never seen anyone more tirelessly devoted and dedicated. I had the privilege of working with Bob in the passage of Prop 14, and his personal commitment of time, money, and talent was unheralded. It was heralded, but unprecedented. So in any event, I think since Bob is the architect and author of Prop 14, it would be great for him to give you a few words on how this 1.5 billion fits in with the rest of the CERM initiative. So Bob, I'm going to turn it over to you.
Thank you, Garen, and thank you, Dr. Freimer and Chancellor Block for hosting this important discussion. as Garen, as you know, the 1.5 billion is really a floor set aside for the brain central nervous system. It is critical that we understand that while this is a major block of funds, it's vital that we have a national and a global collaboration in this area. I've certainly talked to Jonathan Thomas, the chair of the funding agency that I formerly chaired, that was set up with Proposition 71. But this 1.5 billion of the 5.5 billion approved by Proposition 14 is really intended to create a challenge nationally and globally for collaborations in a field that will take an immense amount of money to really make dramatic advances. We certainly understand that the board and the staff of the agency will have to set up their strategic plan that they're currently going through, where they're focused on this set aside for the brain. And we know that there is an entire history of collaborating internationally when I was chairman, certainly, of that funding agency, so that there is a real tradition to work with here in really working with the Healthy Brains Global Initiative and other initiatives like the parallel efforts by the National Academy of Medicine. I put a directive in the initiative when I wrote the initiative that the agency should optimize funding leverage, including national, international territories and institutions, so that it would bring together the best and brightest from around the world. There's a history here that's been very productive in international collaborations, like the effort you are leading, Garen, and Victor Zhao is leading with you. If you look at 2008, you see that the California agency set up collaborations in that year with Canada and Australia, with an MOU that resulted in a single integrated review, peer review process with recommendations to the board. That effort led to, for example, some extremely effective work with Katrina Jameson, Dr. Katrina Jameson from UC San Diego, teaming up with John Dick, and Dr. Slayman from UCLA teaming up with TACMAC of Dr. TACMAC from the Canadian side. Those two teams came up with 4 different human trials that successfully went forward, and a pre-diagnosis and discovery that would lead to early intervention in a deadly form of leukemia. They've now led to what appears to be 3 therapies that are going to be approved by the FDA. Tremendous productivity because of the sharing of knowledge. And we've seen certainly recently with Jennifer Doudna, her collaboration between UC Berkeley and Germany and Sweden in the patents that were developed on CRISPR-9, how effective international collaboration and how necessary it is. So we think that through MOUs and RFAs, that working between your initiative and the state funding in California, you can really leverage the efforts and knowledge globally. Certainly right now, the NIH has a collaboration with the state of California, where the state of California leads the effort on a joint MOU in funding of sickle cell research and clinical trials with funding from the NIH. So there's plenty of effective precedents here, and we applaud your effort and your leadership and this conference among many that you are leading to drive this effort forward.
Thank you so much for that. And setting the stage for us and putting into context how this 1.5 billion fits in with the overall initiative. You set the stage when you talked about the need for global collaboration. So I'm going to spend just a few minutes to talk about HBGI, the Healthy Brains Global Initiative. But before I do, and Josh Gordon is going to follow me to really dive deeply in on neuropsychiatric research. But let's just set the stage for the cause. Can I have the next slide? Many of you will know this data, but could you advance the slide for me, please? So this is an urgent global need. It's the biggest unmet medical need. It's the #1 cause of adult disability. 450 million people live with these illnesses. I think the number many would say is closer to 1 billion. And the burden is $3 trillion annually. The World Economic Forum estimates it's going to $6 trillion by 2030. And as we measure it in terms of dailies, daily adjusted life years, it's the #1 cause of adult disability, way higher than cancer, diabetes, heart disease, and yet we are dramatically underfunded relative to this cause. May I have the next slide? If we bring this now just to the state of California itself, we just recently seen estimates that by 2030, the cost of mental illness in California itself will be approaching $420 billion annually. Wow, if that isn't a reason for us to get started and work together to manage and make a difference in these causes, I don't know what is. My sources for that are at the bottom of this slide. May I have the next slide, please? So in 2019, we launched the Healthy Brains Global Initiative with the support of the World Bank, the World Economic Forum, the World Health Organization, and UNICEF, and working with advisory capacities with OECD and the NEH. We have been working over the last 2 years to build an umbrella set of sustainable bonds and funding mechanisms to deliver evidence-based interventions and brain science breakthroughs and bend this $3 trillion global cost curve. If you think about a 15-year life and a $3 trillion cost at $45 trillion, there is no way this $10 billion won't not only pay for itself, but much like the CIRM 1.0, which demonstrated a 5-to-1 return, we believe we'll be able to demonstrate equal returns. And this 10 billion may in fact be just a demonstration project of how far we can go. Next slide. As Bob highlighted, this is going to work only because we are going to innovate and collaborate. We're not going to do what others are doing. We'll build on the success of others and do things that others are now not doing. Things such as global cohorts, living labs, and a data harmonized major data sharing platform, which we're calling the Information Commons, is going to allow us to, for the first time ever, bring the global neuroscience community together, to be able to not put all the data in one place, but be able to federate information and data sharing across multiple countries. And it's the reason why people will join, because even $1.5 billion, while incredibly significant and a watershed level of funding, is not enough for us to crack the code on unlocking the mysteries of the brain. And by sharing and collaborating and leverage our information across this global platform, we're going to go faster, avoid tuition fees, and build on success. Next slide. This is where we are right now. We are focusing on adolescents and youth, and we won't bore you with how we got there, but we're going to start with depression, anxiety, schizophrenia, bipolar epilepsy, and traumatic. Advance the slide, please. Where by July, we'll have actual clarity on 2 conditions in countries that we're going to go with, clarity on the initial focus, and starting to work on the care continuum. And next slide, please advance. By October, if you advance one more time, please, we expect to actually have clarity on focus and actually begin to be letting RFPs for such research. Next slide, which is my final slide. So the 1.5 billion of CIRM funds is incredible. Our goal with HBDI is to establish a research collaboration and framework. As I said, the California burden by itself is going to be 420 billion. What I don't say on this slide, but all of you know this, that California itself is either one of or the highest concentration of neuroscientists and neuroscience talent in the world. And we hope to leverage that talent in collaboration with the research going across the world. And again, as the number 5 economy in the world, we hope we can allow California to be the lead collaborator and drive global change and positive ROI for California and all who suffer from a mental illness. Nelson, thanks for the chance to open this wonderful symposium. I'll turn it back to you.
Thanks very much, Garen. Now I'd like to introduce our next speaker, who will set the frame for the rest of the symposium. Dr. Joshua Gordon is the director of the National Institute of Mental Health, and he'll talk about stem cell research, genetics, and reducing disparities in mental health, and why they're important to the National Institute of Mental Health. If you have a question for Dr. Gordon, please post it in the Zoom Q&A function during his presentation, and then I will be asking him questions at the end. Dr. Gordon.
Thanks, Nelson. And if I could have the slide presentation, please. I'll take you through it. So while we're waiting for that to come up, by way of introduction, I am the director of the National Institute of Mental Health, which is the principal federal agency responsible for funding mental health research. Do we have the slides? I don't see them up yet. Here it comes. Excellent. All right, next slide, please. We support some 3,000 grants and contracts throughout the United States and indeed throughout the world. We also have an intramural research program that supports approximately 600 scientists working on the NIH campuses, mostly in Bethesda, Maryland. Next slide. Our portfolio is broad, and I'm only going to have a chance to talk to you about one tiny slice of it. But I think it's the slice that's most relevant to the discussions today, although I'm happy to take questions about other aspects of the portfolio that you might think relevant. And I'm going to start with one tiny slice of that tiny slice, which is the progress we've made in the field of genomics. And I think you'll understand why I'm taking that approach as the talk goes on. So this is a so-called Manhattan plot. Most of you will be familiar with this. Of the risk loci, the places in the genome that are associated with increased risk for, in this case, schizophrenia, although the plot would look similar, if slightly less populated, for any of the mental illnesses that we've now established to a certain extent the genetic basis of. And the important point of this slide is that there are hundreds of these green diamonds illustrated at the top of each of these skyscrapers in this Manhattan plot. There are hundreds of loci within the genome that increase risk for schizophrenia. Next slide, please. This, even that complexity, I'm sorry, we should move forward again. Even that plot minimizes the complexity of schizophrenia because the hundreds of places in the genome that are linked to schizophrenia are illustrated on the right-hand side of this plot in blue. And on the left-hand side are yet several dozen more places in the genome that are linked with schizophrenia. And the point of this slide is that there are two different types of genetic risk for schizophrenia, just like there probably are for most other mental illnesses. These rare genes that cause pretty substantial increases in risk for schizophrenia on the upper left, and these more common genes that I was showing you in the previous slide on the lower right. So not only are there lots and lots of places in the genome which can be linked definitively to schizophrenia, but there are also different kinds of these risks which pose increased or decreased levels of risk depending upon what gene you have. Next slide. Now, of course, in addition to the genetic causes of mental illness, there are environmental contributors, and many of them are illustrated on this slide. I'll point out that the bottom one, family history, is the sum total of your genetic liabilities, and it is, of course, the largest of these risk factors, but there are many substantial risk factors, including exposure to famine, exposure to urbanicity, stresses in the prenatal period, including both infection and psychological stresses, stresses in the obstetric period, that all raise your risk for mental illnesses and for schizophrenia in particular. So how do we understand this risk is an important question, which I think we need stem cell technologies in order to be able to address, as I'll show you in a minute. But also, and importantly, and this slide illustrates this fact, these risks are not distributed equally through the population. And unfortunately, most of our knowledge, especially in the genetic side, comes from a rather limited slice of the population. Next slide. So this brings me to my 3 points that I hope to make in the remaining time that I have for you today, in terms of what we are trying to do, both at NIMH and really throughout the mental health research community, to address this success in genetics and turn that success into success improving care for individuals with mental illness, and I should emphasize for all individuals with mental illness. The first thing that we need to do is increase the diversity of our samples to really truly understand the genetic basis of our illnesses for the entirety of the US population. The second is to translate these causes into neurobiology so that we can better understand our illnesses. And the third is to take that neurobiologic knowledge and push towards treatment and prevention. Next slide. So let's talk about diversity for a moment. Knowledge of how these genomic variants vary by the population is very, very clear. Our genomic knowledge about risk, say, for schizophrenia and other mental illnesses is restricted, frankly, to the populations that we've studied those illnesses in. And unfortunately, those populations are overwhelmingly white and European in origin. So in order to understand how these genomic variants contribute to disease, we need to diversify the genetic data. The second, of course, is in order to be able to produce scientific results that will benefit individuals who aren't white and of European descent, we need to be able to understand all the factors that contribute to schizophrenia and other mental illnesses in minority populations. I should say US minority populations, of course, but throughout the world, these are not minority populations at all. Next slide. One quick pictorial example of how genetic information does not generalize from European ancestry to other ancestries is illustrated here, which is a graph of the prediction accuracy based upon genetic information and the likelihood to get 17 different quantitative traits. These are not psychiatric traits, but the analogy holds. If you understand information from a pure European ancestry, and you can predict the likelihood to have high blood pressure or height or various other quantitative traits, you only can predict about half of the likelihood in the broad American population, slightly less than that in the South Asian population, even less again in the East Asian, and dramatically less in populations of African descent. And so, again, your knowledge about a given trait and its genetic contributors is limited to the populations in which you've studied it. Next slide, please. So in order to, go back one. In order to understand the true genetic causes of mental illnesses, we need to develop samples of individuals with increasing diversity. So we've devoted now most of our resources in the search for additional genes in mental illnesses to ancestrally diverse populations. These are two examples of studies that we have funded: South African Xhosa and Latin American and African ancestries, of relevance, of course, to minority populations in the US, but they are, of course, majority populations throughout the world. I would make one additional comment that might be relevant to you all here today, and that is that even with NIMH's considerable budget, and we're pushing about $2 billion a year, we can't afford to cover all of the relevant populations in the US, in the world that are relevant to the US population. And so we look for help and partners wherever we can get them. Next slide. So that's the first piece. We need to increase the diversity of our samples. But once we have the genomic data, once we know what genes contribute, we need to then understand the biology that those genes implicate and how that interacts with these environmental variables, which of course also differentially affect different slices of the US population. And that involves, starting on the left-hand side of this graph and moving all the way up into the upper right, a process of understanding biology at the level of genes and molecules, through the level of cells and circuits in the nervous system, which are of course the tissue level biology we're talking about, through to the outward manifestations of the of function in these lower systems in behavior and in clinical syndromes. And so NIMH has embarked, next slide, on a range of activities that seek to understand these different levels of the biology of the nervous system and how genes and environments interact to alter the structure of genes, to alter the function of cells and the function of behaviors. And in gray here is really what's central to our efforts in the stem cell arena that I'll get into in a moment, which is to try to develop human tissue-based, high throughput, scalable technologies and assays that will allow us to, in humans rather than in animal models, understand the interactions of these many hundreds of different genes and many different environmental factors, and understand how they affect the developing nervous system and eventually the behavior that that nervous system alters. Next slide. So how do stem cells come into play? On the left-hand side, I'm showing this. Gordon is no relation. On the left-hand side, I'm showing a plot, and I believe Sergei can correct me, but I believe it's from his lab. But if it's not, it's from a collaboration that NIMH funds, looking at human cells grown in a dish, how they age over time, and how that correlates with data from human fetuses to understand that we can actually study different periods of nervous system development using human stem cells. So on the x-axis is a bunch of different developmental stages from human tissue that's developing in fetus through development, and on the y-axis is the age of human cells grown in so-called spheroids. You'll hear more about them later in a dish, and you can see that over the first 300 or so days of development in a dish, you have high correlation between gene expression on the x-axis in fetuses and on the y-axis in the in vitro tissue. And at about 250 days, that switches over. So now that you start having high correlation to later developmental stages. So we can study through the course of evolution in a dish or development in a dish, we can study relevant molecular processes, at least, to what's going on in the developing human fetus. And then on the right, you're going to hear more about this later, but are sophisticated experiments that we can look at to look at the physiology on the top and in the middle, connections that develop between neurons, and at the bottom, the function of those connections as these spheroids develop in a dish. So we can get quite a sophisticated understanding of the molecular, cellular, and circuit components of the human nervous system by studying cells in a dish. Next slide. The problem is, of course, that this is a rather expensive and slow process, and we need to find high throughput ways of studying these hundreds of risk factor genes in these cell systems. And one example is shown here. This is another NIMH-funded project that uses organoids composed of lots of different donor cell lines and is able to tag each individual cell and study the molecular components of those cells, and therefore look at genetic variation across populations using even small numbers, relatively, I should say, small numbers of these organoids in a dish. And it's these kinds of high throughput technologies that we need to develop and study if we are to break the translational code between genes, environmental factors, and eventual expression of behaviors relevant to mental illness. Next slide. And what's the end goal? The end goal is to translate that knowledge we get from cells in a dish into cells in the actual nervous system. And here, again, NIH and NIMH are investing in technologies that allow us to study many, many thousands and hopefully millions of cells in single cells in human brain tissue to understand their molecular makeup, their connectivity, and their function, so that we can apply the knowledge gained from these cell systems in vitro to the intact human brain and understand potentially all the way through to behavior, what these gene alterations do. Next slide. So what are NIH and NIMH doing? We're building the tissue and data repositories necessary to be able to understand the effects, and to be able to test and develop, test these genetic and environmental interactions in cell systems with tremendous diversity. We are funding the technology approaches that allow these genes to biology approaches, both early stage and also potentially startup, scale up technologies. We're building knowledge catalogs, both through the BRAIN initiative using maps and also through our own initiatives doing gene expression and omics on large numbers of single cells in the human brain, and building tools for tissue manipulation so that we can eventually test the knowledge we're gaining in intact systems, and then hopefully even use that information to develop cures and treatments. Next slide. But what are we not needing? What do we need from the community? We need increased efforts to recruit diverse samples and explore gene environment interactions in those samples. And that's going to require lots and lots of collaboration. The second thing we're not doing, or that we need, is the commitment to explore definitive causal pathways from our scientists. We need scientists to focus on these established risk factors from diverse communities. And third, we need infrastructure to be able to carry out this high throughput biology. It's all well and good for us to fund the development of these assays in stem cells, et cetera, but we really need scientists to be able to be thinking and building the infrastructure so that they can repeat these experiments hundreds of times with hundreds of genes to really do what we call genome scale biology in the nervous system. So that's just in a nutshell what we're up to and what we need. And with that, I'm going to turn it over, and you're going to hear more, I think, about each and every one of these approaches throughout the rest of the afternoon.
Okay, thanks very much, Josh. I'm now going to turn it over to Larry Zipursky, who's going to introduce the next speaker.
Good afternoon. It's a pleasure to introduce our next speaker, Jonathan Flint. He is a professor of psychiatry and behavioral sciences at the David Geffen School of Medicine at UCLA. He will discuss the importance of large-scale genetics for stem cell science and mental health. Please put your questions in the Q&A during the talk, and we've set aside some time following Jonathan's presentation to discuss these questions.
Thank you, Larry. So I will be, as Larry said, discussing the importance of genetics, and I'll be taking up some of the points that Josh Gordon made. Indeed, as you'll see, I've even stolen a couple of his slides. So I'm going to start by just making the point, next slide, please, that genetics has been a critical tool for unraveling the mechanisms of neurodegenerative diseases. And I'm taking you back here to 1995 with the identification of families in which genes were segregating in a Mendelian pattern, in this case for Alzheimer's disease. And with the technology that was then available, mutations were then found, which is shown in this figure here. Next slide. Those families provided the genetic material which has been critical for later discoveries, and a resource that's been particularly useful here has been our ability to model those mutations within induced pluripotent stem cells, one of the key elements, obviously, of stem cell technologies. However, we can do a lot more than that. Next slide, please. And more recently, and you'll be hearing more about this sort of technology later on, it's become possible to model genetic mutations within larger conglomerates of cells, such as in organoids. And here I'm showing a figure where you can see, as marked by that red arrow, the characteristic pathological feature of Alzheimer's disease, a plaque. So next slide, please. Given that success, we'd hope that psychiatric diseases would follow in the path of the neurodegenerative disorders. And the first point to make is that psychiatric diseases are heritable indeed, as this slide shows, which is a summary of family and twin studies carried out in psychiatric diseases. Some of them are even more heritable than Alzheimer's disease. So bipolar disorder and schizophrenia stand out as really very heritable conditions. Next slide. However, and most critically, no one has found genes for psychiatric disorders segregating in Mendelian forms. And I'm showing in this attempt to write out the pedigree of, in this case, a bipolar disorder from an early publication when people were trying really hard to identify these families. And I think it's just a simple lesson to take away from this is such pedigrees do not exist. So that meant that identifying the mutations underlying psychiatric conditions had to take new forms. And there's really two ways that have evolved over the last 20 years. Next slide, please. The first one is to carry out a genome-wide association study, and the basic ingredients of this are very straightforward. It's the identification of DNA polymorphisms in the genome and the categorization by genotype controls in cases in the sort of contingency table that I've shown on the left-hand side. And once you've collected that information for very large numbers of cases and controls, you can plot the significance of the association as shown on the right-hand side. Each of those little blue dots represents the results of carrying out an analysis at a single DNA polymorphism. The vertical axis here is the likelihood scale. Next slide. So this is one I've stolen from Josh. As you can see, this is the picture you saw before for schizophrenia, deeply populated in this case with more than 200 risk loci. And I've added an extra bit of information at the bottom of the slide, which is the large number of individuals needed to carry out this study. A whacking 160,000 people were recruited into that study. Next slide, please. The second method that's come to the fore for identifying loci involved in psychiatric disorders is really exome sequencing. So here, the idea is you sequence the coding regions of the genomes of large numbers of individuals, and then you treat each exome, each gene as a single unit. And the plot is exactly the same as the one before. Again, there's a vertical axis that gives the likelihood of the association. But now each of these dots represents not a polymorphism, but a signal attributable to variation within a whole gene. And you can see that above the red dot line, which represents the significance threshold, there are about 10 genes which the investigators here think are significant. And again, I've added at the bottom of the graph the numbers of individuals. And again, you can see this is a huge number. There are 70,000 genomes approximately had to be sequenced to generate this level of security of information. And clearly many more could be identified, as you can see that the dots continue downwards. We're not there yet in terms of identifying the full amount. Next slide, please. So again, this is the second slide that I stole from Josh, and I want to make just a couple more points about this. So the horizontal axis represents the frequency in the population, and you can see on the right-hand side where we're looking at variants that are maybe occurring in 40–50% of the population, that there are many loci that contribute, but they're all a very tiny effect. So the vertical axis, the odds ratio, if you have an odds ratio of 1, there's no effect. Anything above increases your risk or below decreases your risk. And you can see the vast number of loci all congregate really close to 1. On the left-hand side of the graph, where we're dealing with much rarer things, down to, as you can see, 1 × 10⁻⁵, really tiny frequencies in the population, there are large effects. Ignore the green dots, the copy number variants, which represent relatively large rearrangements. The important thing here is to look at the red dots. And note that these genes, these red dots, even though they have relatively high effects, they're not at really 100, greater than 100. Even if you have one of these things, you're not definitely going to get schizophrenia. It's just an increase in risk. It's still an increase in relative risk. It's not like a Mendelian effect. Next slide, please. So just to summarize what I've said. So psychiatric disorders are heritable. They're highly polygenic. Thousands of risk loci contribute, and almost all of that risk is contributed by those common variants of small effect. And those common variants are almost always located in regulatory regions of the genome. The rare coding variants, which have relatively large effect, exist and are now gradually being discovered as we run ever increasingly large exome studies. Next slide, please. So what are the implications of the genetic architecture for modeling disease of psychiatric disorders? And there are 2 points I want to bring to your attention. And the first is given in the next slide, which is that because we have so many loci contributing and each is making a small contribution, it's going to be really hard for us to disentangle individual effects. And that means we're going to have to work close with computational biologists to analyze genetic networks. And that, of course, is a study that's well underway now in groups of people who've been looking at gene expression and other components of omic regulatory information. But the analysis of genetic networks is clearly going to be important if we're going to take forward the discoveries from the genetic work that I've described earlier. Next slide. And the second thing, which again follows from the complexity of the genetic architecture, is that genetic action likely involves multiple cell types. And restricting our analysis, therefore, just to using stem cells is unlikely to give us a full picture. We are definitely going to need to look at organoids to model in a far more sophisticated fashion the actions of these genes in multiple cell types within tissues. Next slide. So the examples that I've given you have been primarily from schizophrenia, and schizophrenia, bipolar disorder, and autism are the success stories in genetics. And we now want to move to look at other conditions, drug abuse, anxiety disorders, and major depressive disorder. And I'm going to choose major depressive disorder because that's what I've been working on, but it's characteristic in many ways of these other conditions. And I'll just give you a brief overview of what those characteristics are. Next slide. So this is a common condition, commonest cause of disability in the world. It's associated with mortality through suicide. And the last 2 points are perhaps the most critical. There is a large effect of the environment. So although it's heritable at about 40%, we can't ignore environmental effects in this condition. And also partly because of this, we're pretty certain that there are many ways in which the condition could arise. It is heterogeneous. Next slide. I'm going to give you one example about how we've been dealing with this problem with the work that we've carried out in a large cohort in East Asia. And we've worked there because we've been working with a particularly severe form of depression. Next slide. So on the left, I'm showing you how I think most people imagine that life events, environmental effects are related to depression, which is that there's something terrible happens in your life. And within a few weeks or months of that, an episode of depression occurs. Now that has been well documented for decades by psychiatrists, and we know that that relationship is very complicated. And on the right-hand side, I'm just giving you one example of how that complication exists. So in that figure, I'm showing on the horizontal axis the number of a depressive episode. So for example, 3 represents the 3rd episode of depression, 4 represents the 4th episode, and so on. And on the horizontal scale are the proportion of our cases reporting a stressful life event. So what you can see here is that when we ask people about their first episode of depression, they are saying that maybe 60% of those first episodes are related to a stressful life event. But people, when they're asked about their 5th or 6th episode, only about 25 to 30% of the episodes are related to a stressful life event. So something has happened during the disorder that's changing the relationship between the environment and the onset of the episode. So how can we explain those sorts of events and take them apart? Well, we can use genetics. And my last data slide, I want to give you one example how we've done that using our cohort from Southeast Asia. So what you're seeing here are 2 of these Manhattan plots, these associations carried out for common variants. And the top Manhattan plot shows an association between depression, which is due to stressful life events. And you can see 2 loci on chromosome 10, those are the red dots. If you look at the bottom graph, exactly the same display, but this time the study is for cases of depression where they are subjects and after multiple investigations can report known cause. And again, we're finding some loci, the red dots, but note importantly that the red dots are in different places. And indeed, if we carry out a genetic correlation analysis, we find that the correlation between these two types of depression is not unity. There is a lot of genetic variation that explains that difference. These are not the same conditions at a genetic level. Next slide. So just to summarize, genetic analysis of common psychiatric disorders is possible and it's ongoing. We are able to deal with the complexities and we are able to find loci. Many common variants have been identified and exome sequencing is in the process of identifying larger effects. These are the sorts of genes that we will be able to use for downstream functional analysis. And it's my last slide. Next, please. The progress in genomics and computational analysis provides ways to bridge disease to cell and molecular mechanisms, and the stem cell technologies, including organized based on patients' genotypes, is a way to study the molecular and cellular bases of disease, and in time promises to provide pathways to therapy. Thank you.
Thank you, Jonathan. I'd like to encourage our viewers to place any questions they have in the Q&A. And we have one first question from Lila Collins for Jonathan. Could episodes of depression leave an epigenetic imprint? Perhaps this leaves individuals more at risk for more episodes.
Yes, that is one way to interpret the data that I showed you, to understand how, when someone has had one episode, they seem to change the way they behave the environment. Our interest, however, at this stage is really to understand what the genetic effects are, because I think those are the ones that we're most likely to be able to model using current stem cell technologies.
So other questions? So I will ask one and give people time to scribble other ones into the Q&A. And this sort of takes off from your last slide, perhaps, and that is, what do you see as the major priorities for basic neuroscience to use polygenic data to uncover molecular mechanisms?
I guess you're talking here more generally, not just about stem cells, Larry. So it's
no, just generally. Where would you go? I mean, one is you summarized on your last slide, sort of computational approaches and stem cells, both, I think, powerful ways to approach these issues. Do you think there are other ways to do that you've considered?
Yes, these are just one of an armamentarium of resources that we should be bringing to bear. Josh Gordon mentioned some of the other ones. There's clearly a lot that we need to think about in terms of model organism work. What are the most suitable ones? What are the type of manipulations that we could employ? On top of that, I think we really need also to consider in a bit more detail what type of phenotypes we really want to explore. So far, the analysis has been rather restricted to things like schizophrenia, the psychosis. How do we imagine we're going to get information around delusions and hallucinations? How do we factor all of that in? So I think from a very large number of disciplines, we're going to have to draw upon a lot of people to come up with some ideas of how to push this further forward.
Great. Let me turn to some additional questions from our audience, from Michael Gorin. Have you considered using Mendelian randomization approaches to consider tests of causality of other factors with mental illness?
That's a good question. I and many other people have indeed adopted those approaches in order to try and understand the relationship between different disorders and depression. There's an entire talk that we could give about that. The short answer to that question is yes.
Another question then. Let me take a quick look here. From Hua Chai, are you able to follow patients after initial MDD episode with known stressful life event to study changes in their epigenetic or genetic expression when they have a recurrent episode?
That's a great study. We'd love to carry it out. That type of longitudinal study is what's needed at this stage, but it's going to take a lot of money and a lot of time to set that sort of thing up. But yes, I'd love to do that.
We have one from your colleague, Carrie Bearden. Do those genetically distinct subtypes of depression otherwise differ phenotypically or in terms of biology?
Yes, they do differ phenotypically as far as we can tell. They differ, for example, in the fact that those people who report without any known stressful life event seem to have a more, what I would call vegetative type of depression. In other words, they're reporting more biological features, disturbance of sleep and of appetite. There are other features as well, but I think those are the most characteristic.
Let's take one last question from Jeff Lomax. When referring to environmental exposures, I assume physical, psychological, environmental. Can you elaborate? Specific interest in how environmental pollution monitoring might inform environmental exposure association.
So most of the work that we've done is by interrogating people about what we know to be important in the environment, and that's mostly to do with stressful life events, parental relationships, their background, how they've been brought up. But it would certainly be interesting to look at the other features that you mentioned. We haven't done that yet.
Some evidence to suggest links between pollution and autism and among other illnesses. And there is more and more interest in looking at things like matching geography and various social determinants of health to a number of these different illnesses.
Great, thanks, Josh. I think we should move on. Thank you, Jonathan. Nelson will introduce our next speaker.
Thank you, Larry. So now turning from genetics to populations, I'd like to introduce Dr. William Vega from the University of Southern California, who's going to talk about the impact of health disparities in mental health. And again, just to remind you, please put any questions you have in the Q&A to be asked at the end of Dr. Vega's talk. Go.
I just want to compliment the previous speakers because they've set a really great opportunity for me to explain things that I'm glad I don't have to do the background story for some of what I'm presenting because of time limitations. I'd like to move ahead now past the first two slides and to slide 3. I just wanted to start here because I think this is an exemplification of people who are under extreme duress that brings forward many of the things that we're talking about, of all the factors associated with the expression of psychiatric symptoms and disorders. These are Haitians and actually who are trying to get from Haiti to Central America, Mexico, and we're stuck in Tijuana in a refugee center trying to get through. And I don't know if they ever succeeded or not, but you can see the conditions under which many people have to struggle, and has a lot to do with what you end up seeing clinically in the worst of circumstances. Can we move forward to the next slide, please? So the aims of my presentation are really 5 things I want to feature as key issues. Latino health and mental health disorder patterns have implications for advancing knowledge about the role of genes in mental health. And I think that California provides an excellent example of that because we have such a large population. There's lots of diversity in it. There's immigrants, there's US born, and they have vast differences. And of course, they immigrate across a border where there's an entire nation that we can study and compare. Mental health rates vary greatly in Latino immigrants and native born, and understanding these differences has salience in thinking about causal mechanisms. The personal impact of intergenerational and social adaptation to US society has a profound effect on behavior and mood disorders and substance disorders, and sustained low education and income set a foundation for propensity towards a set of experiences and health threat exposures. And ultimately, risk behaviors of Latinos and the conditions they're exposed to are reciprocally related to generic liability of psychiatric disorders. And I'm going to give you some data and information on that as we proceed. Next slide, please. I just want to say that this is not a random situation. That's why disparities researchers look so much at location. This is LA. And as you can see in Los Angeles, this is diabetes, but it holds true for hypertension, it holds true for depression, and also for COVID-19. If you look at the dark area, it's East LA, Central LA, and the South LA, and the blue area, the darker blue area follows it too. So you look at the yellow area, that's UCLA, the area around UCLA, of course, West LA, and the differentials are just massive. And we see that in everything. It goes from less than 11% to up to 29% in the dark blue areas of diabetes. So that's just one example. And depression also has, those are the big 3. I mean, depression is one of those as well as distributed in a similar way in LA. Next. I believe too that there's a systematic way that we can begin to understand this, because obviously this is not a singular event in which people develop a disease. There's a process involved. The process is really a process of reactivity to the environment that people live in. And when people are born with low circumstances in terms of low educational opportunity and low educational accomplishment, low literacy, low income, and they are really in a stratified situation where you can anticipate that they're going to experience high burden over the life course with employment issues associated with more toxic exposures and unemployment and over again, life chronic stress, unhealthy behaviors, low healthcare access, diet and exercise deficits, and of course, ultimately, at about the age of 60 or so, chronic diseases, functional limitations, et cetera, onset, including dementias. So next, please. Now, we know that this is the simplest way that we can possibly express this issue, which is the genome sets a stratum for the individual, and that individual goes forward into the world as an infant and then spends a lifetime of circumstances created by the culture and social environment and physical environment that they live in. And the interactions across those are creating phenotypes differentially, and those are, of course, then feeding back to the genome. But this is not linear. This is an interaction that's taking place across all of these levels simultaneously. Next. Now, we know even among primates that if you change their environment, if you put them into situations where they change their social order, or you put conditions of stress on them in other ways, that they experience difficulties in terms of their mood, their behavior towards each other, and in worse situations, even death. So that we can take some clues from that. Moving forward. So this is the fundamental question that I'm presenting. Is the question is whether different genotypes have a different relationship to the phenotype in different environments. And I have excellent data, I think, to exemplify how this takes place. This was something that was put forward by Cooper as a core question. And I would just change it to say, how is it that this relationship change takes place? Because that's what we want to know. How do those pathways alter in the context of social environment interactions in relationship to the substrate of genetic factors that put people at risk? Next. Now, this was a model actually produced by Cooper, which is sort of his own version of a thought experiment, I think. He looked at Africans in Africa, which were the countries of origin for African Americans in the United States. And he said, if you have a gene variant absent and gene variants present for hypertension, what you're going to find is a small difference in that country, in those countries in Africa. But in the United States, what we find is that gene variants absent varies enormously in magnitude from gene variants present in people who have those variants in the African American population. And so in the United States, we see much higher levels, of course, of cardio events and also stroke death. And it's quite different than anything that you would observe in Africa. Next. Now, I want to speak to the context of Latinos in California and in Mexico specifically, because they give an excellent example that we're up close to. There's two sides of the immigrant health paradox. And I mention this because health is very related to mental health, of course. And we see that, for example, that the health of immigrants is actually better than it is of the US-born Mexican Americans or the general US population. Latino immigrants of Mexican origin live about 3 years longer than the US population overall, almost 81 years, at least before COVID, and lower death rates for heart disease, breast cancers, and lung cancers. They have healthier babies, and they're much less likely to have smoking dependency, alcohol dependency, or other type of drug dependency. Nonetheless, by the time they're 60 years of age, this whole picture changes radically, and we see an enormous deterioration at that point, very high risk of disability, much higher than the US born. And so there's a real change over the weathering that created by the life course, because obviously this is a population of extremely low income and education, and suffers from a number of marginalities, including healthcare access. Next. So this is based on the work that I've done and Ron Kessler did, and really other people as well over the last 20 years. This is based on the data from the National Comorbidity Survey, if you look at the pillar on the extreme right. And this is actually the figure for lifetime prevalence of a DSM disorder. And you can see when you look at the data as compared to Mexico City, for example, that 48.6 is the rate of total disorders. In Mexico City, it's 23.4%. And in Mexico, it's very similar to immigrants in California. Mexican immigrants in California had 24.9% and native-born at 48.7%. Literally double the rates, 100% increase in rates occurring just by crossing that border. And even in the United States, these immigrants are still retain some of those residual protective factors and are less likely to have a sense of reactivity to the factors involved in their lifestyles in the United States. Next, please. Now, this was a model that was given to us by Nora Volko that she used for several years in presenting her work, Systems Operating the Etiology of Drug Addiction and Mental Illness. And I use it only because, we, of course, are concerned with the very high interactions between mental illnesses and drug abuse and dependence. And this creates further consequences, it seems to me, for trying to discover triggers, for mental illness and for drug abuse and for addictions, because obviously all of these stratum that we see here of genome, protein expression, and so forth, we all understand those, but the complexity of the pathways, the actual causal chains that develop to actually influence these is very important, because we know people with schizophrenia and bipolar disorder have extremely high rates of drug addiction, which is true in the United States and not true in other countries, which I think this is extremely important to bring forward. So we're looking for things that are occurring in this country, in this social context, and we're looking for causal factors, but it's a difficult thing to explain it when you look at Central America or Mexico and you get bipolar disorders and schizophrenia with no or marginal use of substances and addictive substances especially. Next. So let's push forward 2 slides, please. Next one, right there. This is an example of that. You can take a look at that and you can see that this is from the national survey that was done of Latinos in the United States. It was a study funded by the National Institutes of Mental Health, as all the previous work was as well. The alcohol abuse or dependence, with or without a drug dependence and co-occurring non-addictive rates, total for immigrant women was 0.68%, less than 1%. For men, it was 5.25%. These are immigrant men and women, Mexican origin in the United States, actually in California. Total for US born was 7.33% and men 16.22%. So you can see these vast differences in the risk factor that the drugs in a society really represents. And it's not because these societies, like, for example, like Mexico, don't have access to those drugs. And it's not that these immigrants in the United States don't have access to those drugs, it's that they're not going to use them. And we needed to make I think a discovery of what is the difference in the likelihood that the risk factors of interest, including the genetic substrate, are influencing differentially these different groups. Next. I'm put this forward only for one reason. This is actually the 12 factors that were put forward in the Lancet 2020 report associated with preventing dementia. I mention them only because the factors are all, for the most part, except for one, I believe, are all associated with heightened depression. And so you look at diabetes and smoking and air pollution and obesity, et cetera, and depression itself. And of course, we know that this operates through pathways and ultimately increases the risk of dementia. And I think there's an important lesson here because the lesson here was that 40% of dementia could potentially be decreased. And in Mexico, they actually thought it would be more like 56% of dementia. So I think these factors, lowering the risk, the presence of these risk factors in society, which is a public health issue, has a lot to do with helping us deal with the problem of the interactions potentially with high-risk genes for mental disorders. And it's obviously going to be much easier for us to deal with certain types of disorders that are autosomal, for example, or more likely to be high heritability than it is to deal with things like depression, anxiety, which are the more common disorders, and because they're more episodic, but also because we need to know how to lower the likelihood that these things are going to be recurring over time. And it seems to me that anything we can do to mitigate and decrease the likelihood of the disorder at all, but also to be able to more successfully keep it from recurring. Mexican women in California are one of the most highly likely to have continuous relapses of depression. Even though the rates are somewhat lower, they still have continuing relapsing at very high rates. Next, please. So in summary, we say prevalence of common behavior problems vary from society to society. Rates of psychiatric disorders vary across nations and among subcultures. And even behavior traits that are normally regulated, impulsivity, sensation seeking, risk taking, we generally think of those as traits that are really hard set, but they are not, as we have found in behavior in comparing young adults, for example, in Mexico and the United States, and vast differences in impulsivities and risk taking. And illicit drug use initiation and dependence is highest in the United States of all developed nations. Now, can we move forward just a few more and I'll stop there. Right there. The trends in the United States in mental illness for Hispanics are clear. They're rising steadily for serious mental illnesses across all age groups. They're rising steadily for major depressive episodes across all of these age groups. And we know that people are not getting treatment. And it's about 56.8% of Hispanic young adults, 18 to 25, did not get treatment. And there's very low detection of chronic disorders, with 90% of Hispanic people over the age of 12 with a substance abuse disorder not receiving treatment. So these are all risk factors, of course, because they will interact with the genes that they're associated with expression of various types of mental illnesses, especially for depression and anxiety and other forms of, and it puts them into lifestyles where they're more likely to have a lot of stress. So I'm going to finish with, move forward, please. One more. I'll finish with this one. This is a study that I did about 20 years ago and then followed up about 10 years ago. This study was done with kids that were in middle school between the ages of about ages of 13 and about 15 years of age. Over the 3 years we studied them as adolescents, and then we followed them up as young adults. The point here is to look at the impact of life events. Now, the x-axis on this is the cumulative adversity count, the different kinds of different adversities they had encountered cumulatively. And this is the conditional probability of drug dependence by the age of 18. Now, I think it's very interesting for two reasons. First, because half of the reasons that this occurred, that is, half of the actual adversities that were experienced were early in life, in their early adolescence. The other half were much later in early adulthood. And they both had equal predictive power for a drug dependence. And you can see that this is very linear for Latinos and for non-Latinos in this instance. However, it is not linear for African Americans. And we find this in all of our studies. They have lower likelihood of reactivity to depression from these types of adversities, but they have much higher likelihood of having hypertension and having other kinds of things that are going on around metabolic dysregulation in adulthood. Also, the other thing we find with African Americans that is predictive of substance dependence and depression is violence events directly associated with gun use, knife use, being in the situation, being a victim of this or perpetrator or witnessing it. Last, please. And I'll finish here. How much can we really improve mental health and physical health of Latinos using therapeutics without a corresponding improvement in education and income? How can we identify critical mechanisms of genetics that provide information for effective life course interventions? And how can genetics research in mental health include a public health perspective? Does it begin with a common language? And I think that we have a long way to go, but this kind of research is extremely exciting, I have to say. I'll just conclude with this one statement, which I found, and I do Alzheimer's research now for the last 10 years. You know that early onset Alzheimer's has a 100% probability of occurring through transmission to the person that receives the polymorphism. However, late onset episodic Alzheimer's does not have that similar relationship. And when I was in the field up to about 4 years ago, we were talking about APOE4 being responsible and associated genes being associated for about maybe 50 to 70% of the likelihood of a late onset Alzheimer's disease onset. Now we're talking about 7 to 10%. That's a massive reduction in the responsibility and the liability, presumably, of the APOE4 gene and associated genes for the disease. So it seems to me that we have an enormous amount of flexibility here in thinking about this and thinking about associated development of causal pathways and causal thinking. I'll stop there.
Okay, thanks very much, Bill. And again, if anybody has questions for Bill, please put them in the Q&A. I'll start out with one, which is, you've presented a lot of compelling data on the impact of different types of adversity, risks, age of onset, and so forth for many different types of mental illness, substance use, and so forth. And as we've already heard a little bit, a big part of the mission of what can be accomplished through the research that will be supported by Proposition 14 is really directed at how to bring the advances from, let's say, this neuroscience research to really benefit all the different populations in the state and globally. And I just wondered if you have thoughts about how research such as yours on disparities can be integrated most effectively in with the research that's going to be conducted, let's say, in neuroscience, genetics, and stem cell science itself.
Well, I would reiterate that I think that there's two critical issues. One is the inclusion of numbers of Latinos and African Americans and other groups that are well represented in the society that are adequate for study in the research so that you can generalize. This has been an enormous problem in Alzheimer's research as well, that people simply don't include it and they believe it's not really important for discovering the mechanisms. It's very important because we know that these genes do operate differently in terms of rendering risk for these expressions of psychiatric disorders or Alzheimer's disease. So it's critical that we have large enough numbers and support these studies at a level where they can do this. Otherwise, you'll be asking the same question 10 years from now. Obviously, international research is a piece of this. It's very straightforward. I think we have to go into other countries because you're going to find different things and you're going to find new founder genes, et cetera, and new clusters. I think the other thing that is critical to look at in all this is biomarker differences and the value of biomarkers and its predictive validity, for example, in different populations for different aspects of the research. Those are two absolutely fundamental things to move this forward.
Okay, thanks very much, Bill. And now moving on to the next talk, I'd like to introduce Alicia Martin from the Massachusetts General Hospital and the Broad Institute, who's going to follow up that lead and talk about the importance of inclusivity of diverse populations in genetics research as a means to reduce health disparities. Alicia.
Thanks so much, Nelson. And I'm really delighted to be here talking about this topic, which I'm super passionate about and feel like I'm in such wonderful company on this topic. So to get started, I don't need to tell any of you, but I'm just going to remind you that California is the most diverse state in the US. It's a majority minority state with over 39% of the population being of Latino descent or identifying as white, constitutes like 36%, 15% are Asian or Pacific Islanders, 6% African Americans. So this is an incredibly diverse state, and more than 10 million are foreign born, coming from countries all over the world. This is more than any other state, both by number and by percentage. And just like the coronavirus pandemic hasn't been felt evenly, the burden of mental illness is also felt unevenly across racial and ethnic groups. I didn't include prevalences here for various different disorders because they would tell an incomplete and potentially misleading story. So for example, clinicians on average overdiagnose schizophrenia and underdiagnose mood disorders in African Americans compared to other populations. So I think these nuances are really hard to share with just a table with numbers. So our job is really to ensure that our advances in neuropsychiatric disorder research is improving health outcomes equitably rather than widening already existing and massive disparities. Given how diverse California is, it's important background from a genetics perspective to think of this through an evolutionary and historical lens. So as a geneticist, I like to start at the beginning. And for our species, that means starting over 100,000 years ago in Africa. As humans migrated out of Africa, they took a subset of genetic variation with them, which means the average out of Africa population has about 1 million fewer genetic variants than the average African population. So this human history over the course of the past several 100,000 years really informs the landscape of genetic variation that we see across the world and is therefore really important to consider when we're designing these big genetic studies of any disorder. With that in mind, I think it's the most exciting possible time to be working in human genetics. Our studies have grown totally explosively. I've been lucky to watch the field evolve from including hundreds or perhaps thousands of individuals in genetic studies from around 2006 or so, to now routinely including millions of individuals in a given genetic study. So we've learned a ton about human history, like I was alluding to, but also a huge amount about the genetic and biomedical basis of disease from this exponential growth over the past decade. I see genetic studies as really serving the mission of providing biological hypotheses from these unbiased associations and serving as the basis for downstream functional analysis and hopefully eventually therapeutic insight. So we have sort of two big issues with our current sort of status quo in genetics, though. We have this massive diversity problem. So if we want to understand the genetic basis of disease, we actually need this huge amount of genetic diversity because if we have less genetic variation, we're determined to then find fewer genetic associations. However, about 80% of our participants in genetic studies have primarily European ancestry. And this is far out of step with the global population, where European ancestry populations make up about 16% of the world's population. And maybe even more problematically, if you look at the bottom part of this slide, you can see that we're not on a trajectory to correct these imbalances or make these genetic studies equitable across the world and also within our country. So around 2014, our genetic studies peaked in terms of diversity, and since then have perhaps stalled or even slid in the wrong direction for diversifying progress. So we've got a lot of room to grow in terms of diversifying progress. And as I mentioned, with this big need to have a lot of genetic diversity in our studies to ensure equity as we're trying to translate these, this is a major, major issue. So one question that I really like to focus on is how do these study biases in genetics then impact the generalizability of our knowledge? So I think there's really two big important considerations here. The first is that biology tends to be shared across populations. The fundamental pathways that we're learning about that inform risk of schizophrenia tend to be the same pathways across all populations. However, what we're also seeing is as a consequence of this difference in human history, frequencies of genetic variants may differ across populations. As Dr. Vega eloquently described, environments can vary substantially across populations and many other factors can differ. So we may have a pretty big issue with generalizing even if the fundamental biology is the same. So when we run genetic studies, we get DNA from many individuals. And one tool that is becoming increasingly important in our field is a polygenic score. So when we do a genetic study of disease, we often have patients and non-patients. In this case, we might have a cohort of participants in a study that have a psychiatric disorder and that don't have a psychiatric disorder. So we can divide them up into these different groups and then compare their DNA. So when we scan across their DNA, we can see if genetic variants are more common in patients, suggesting an increased risk of disease for that genetic variant. Most genetic variants contribute a pretty small amount of risk though, so we need a lot of genetic variants to predict risk of disease. As Dr. Flint mentioned, it doesn't tend to be the case that for disorders like schizophrenia or bipolar disorder that there are any Mendelian forms. Instead, there are tens of thousands likely of genetic markers that are acting in concert to increase or decrease risk. So polygenic scores make use of these thousands or tens of thousands of markers to, in aggregate, form a score that predicts someone's risk from their DNA of having some disease. This works really well for psychiatric disorders because they're so heritable. Schizophrenia and bipolar disorder, for example, are quite heritable disorders, meaning that our DNA has a lot to do with whether we'll have a disorder or not, though it's obviously not the entire story. So we can see these shifts in distributions compared to those individuals who have a disorder versus those who are healthy and maybe tend to have fewer genetic risk variants on average. So there are many areas where we're thinking about polygenic scores. These are really potentially fantastic biomarkers that can be used in fields like psychiatry. Since the brain is an inaccessible tissue, we've been really stuck for a long time with biomarkers and with drug development, while other fields like cardiology and cancer have been making huge strides already from genetic studies. So I think a polygenic score, for example, is a really impactful potential biomarker that we'll be seeing moving forward. So this is an incredibly exciting biomarker. I can't think of any single lab test that can be so predictive across such a broad spectrum of diseases. So thinking of like cancer and diabetes and psychiatric disorders, and the list goes on. We can compute polygenic scores for all of these simply with DNA and from early on, which allows us to then identify new interventions by sort of randomizing using these polygenic scores and testing different treatments. However, what I see as the biggest challenge to implementation of genetics and precision medicine is this vast Eurocentric bias. So when we do these big genetic studies in primarily European ancestry populations, which make up about 80% of our current genetic studies right now, as I mentioned, we see these staggering disparities across populations. So we're predicting European ancestry traits across a bunch of different quantitative traits about twice as accurately as in East Asians and about 4 to 5 times as accurately as in African Americans and African ancestry populations. This is simply a function of who we choose to study. There's nothing innate about a population's genetic risk that we're learning from these types of studies, except that these differences across populations, we're only understanding the differences in accuracy as a function of this vast Eurocentricity in our data. So I think we really need to accelerate our population genetic studies and think a lot more about health equity in the field of genetics broadly, especially in terms of translating this to neuropsychiatric disorders. So we're going to need a lot of resources, which I think Prop 14 is very well set up to sort of build on. We need a lot of new methods, since many of our statistical methods currently assume a lot of homogeneity. We already talked about how California is an incredibly diverse state. We need new data and community resources to address major gaps in our genetic studies, and we need to develop supporting resources for these growing data sets. We also need bigger research capacity. We need to train the next generation of diverse researchers to lead these big efforts in their home communities and in their home countries, where they will better understand the environmental risk factors that may go along with those genetic risk factors. With all of these in place, there are incredibly huge opportunities that'll be a win for all of us with these new resources. So for example, we can identify novel associations. We can identify low-hanging fruit that's present in primarily non-European ancestry populations, but absent in European ancestry populations. That'll help us pinpoint new biology. We can refine our view of causal genetic variants. We can understand, using fine mapping, which genetic variants cause an increased risk versus which ones are simply correlated and kind of along for the association ride. We can prioritize then which variants to follow up functionally, and then use these to test biological function in cells or in animal models. We can then better understand our genetic and environmental determinants of health. And then, as I've talked about, we can also prioritize genetic risk prediction and especially improving accuracy for all populations, and especially underrepresented populations. So to this idea of building research capacity, I'm really passionate about putting my money where my mouth is. And ultimately, our goal of analyzing these increasingly diverse cohorts, I think, needs to come along with some steady improvements in terms of bringing genetics to all populations. So I've worked with some fantastic researchers in sub-Saharan Africa. I'm showing you 3 research fellows from a research capacity building program called GINGER, the Global Initiative for Neuropsychiatric Genetics Education and Research. I've left a link if you would like to go learn a bit more about this program. These research fellows are from South Africa and Uganda, and we've worked together over the course of the past couple of years to show how genetic risk prediction accuracy increases and improves substantially when you include participants with ancestrally diverse backgrounds. For example, including not just European ancestry populations in a training data set, but also Japanese individuals, African Americans, and Hispanic Latino populations, as well as a UK-based population, for example. Across the board for several dozen traits, we're seeing that accuracy is improving, especially in these populations, especially in Hispanic and Latino populations, especially in East Asian populations, and especially in African ancestry populations, reflecting who we studied in the first place. And some traits get a bigger win than others. And that's sort of a reflection of the fact that we're really going to need to deeply understand these disorders and their genetic architectures to understand where there are going to be some big wins here. So I just want to end on this study that I'm incredibly excited to be a part of. And that's a wonderful collaboration with our UCLA partners. So this study is called the Populations Underrepresented in Mental Illness Association Studies, or the PUMA study. This is an incredibly exciting study that is going to be prioritizing genetic discovery efforts in African and Latin American populations. This will hopefully help us play catch up a lot more with all of these major psychiatric genomics consortium efforts that have primarily studied European ancestry populations so far, and will allow us to explore new questions like which genetic variants are transferring well across populations, and which ones do we see as showing more distinct effects, and are we only identifying by studying these underrepresented groups. We'll be studying primarily bipolar and schizophrenia in this study. So we'll be able to also identify which genetic variants are shared versus distinct across these disorders. And we can look at which genetic causes of these disorders are the same versus show any heterogeneous effect across populations, which we expect to be more the exception than the rule. And we'll also learn how biology is perturbed by genetic variants that increase risk here. So this is a big, truly a massive endeavor. This will be building a sample bank of about 180,000 participants across these countries and regions across Ethiopia, Uganda, Kenya, South Africa, Nigeria, Colombia, Brazil, and parts of New York and Southern California. This will be, as I mentioned, in partnership with collaborators at UCLA, of course. Through this exciting activity, we'll be sequencing the genomes of 120,000 participants, including about 40,000 with schizophrenia, about 40,000 with bipolar disorder, and about 40,000 population matched controls. Notably, this will be a sustainable resource that'll empower downstream genomic studies in these underrepresented populations in genomics because we're doing whole genome sequencing, sort of a durable technology for future studies. And notably, we're building research capacity alongside this big endeavor and returning data to all of the participating sites in this major study. So as next steps, I'm really excited about the potential of genetic studies. For example, polygenic scores can provide an objective biomarker in psychiatric disorders. These can improve clinical models and help us identify risk intervention strategies. But right now, I think they're most likely to worsen health disparities due to Eurocentric GWAS biases. So we really need to invest and make sure that we're developing more diverse cohorts. We're developing new statistical methods. We're building concomitant genomic resources and research capacity to address these huge issues. And these topics are not always directly easy to talk about. These are often sensitive topics that I think are warranting a lot of responsible anticipation of implications of our research, responsible communication. This type of work has been in the headlines a lot lately, so I think it's really, really critical that we partner with other disciplines to ensure that our message is spread carefully. I'll stop there. Thank you.
Okay, thank you so much, Alicia. I will start with a question, which is that at the beginning of your talk, you talked about the incredible genetic diversity and population diversity of California. And then at the end, you talked largely about efforts going on in other countries to tackle bringing greater diversity to these genetic studies. And just wondered if you could say anything about your thoughts about, will that be enough to do these studies in the sort of ancestral populations that the population of California descends from, or to what degree will it be important to do these kinds of studies directly in California to address the diversity of the state?
Yeah, that's a great question. One thing I think that's, sort of maybe paradoxical to think about is that if we just studied the breadth of diversity, say in California, or if we reflected adequately the population of the US, we would still be doing a poorer job of predicting genetic traits and building biomarkers from genetic data in, say, African American or Hispanic and Latino populations, because they have such rich ancestral diversity that we're not going to adequately capture that simply by reflecting the population we have. So it's actually critical that we go into these other countries if we hope to equitably serve the Californian diverse population. So great question.
Okay, I think then if there are no other questions for Alicia, then I'll turn it back to Larry to introduce next speaker.
It's a pleasure to introduce our last speaker, Sergiu Pasca. He is an associate professor of psychiatry and behavioral sciences at Stanford. He will discuss stem cell science in psychiatry. And just to remind you, please, if you have questions, please enter them into the Q&A in the Zoom function.
Thank you so much. It's such a great pleasure to be part of this exciting symposium today and to share with you some of our work, as well as some of the challenges and opportunities in using stem cell models of disease. I'm a physician by training, and the main interest of my lab has really been in understanding the molecular and cellular basis of psychiatric disorders. However, one of the main impediment in doing so and understanding the biology of these conditions has to do with our access to the human brain. And by access to the human brain, I really mean access at the molecular and cellular level during development, as well as in disease states. And the reality is that really for most of its development, the human brain is inaccessible. So if you were to just like look at the development of the human brain, it's very clear that we know quite a lot about the early stages of patterning and self-specification. But as you advance in human brain development, what becomes clear is that the more we advance, the least we know. And for instance, we know about some of the populations that are making up the human cerebral cortex, but we know very little about how the decisions, for instance, are made to generate upper layer neurons or astrocytes, which over the course of evolution have actually changed quite a lot in their morphology and presumably in their function as well. And similarly, we know very little about how cells in the human brain migrate. How are they connecting with others? And the molecular basis of plasticity of the circuits in humans is almost completely unknown at the molecular and cellular level. So that's why for in the last period, what we've been trying to do, and of course, by we, I mean the entire field, is really try to capture, not in total, of course, but in isolation, some of these key cellular processes with human cells in an attempt to be able to develop better models for psychiatric and neurological disorders. And of course, we primarily do that by cell reprogramming. And I know by now, everybody's already familiar with cell reprogramming, but showing you here depicted by one of my patients with autism is the cellular reprogramming process, which involves taking skin cells from patients and then pushing them back in time to look like stem cells, and then really taking those stem cells and guiding them to become various cell types in the developing brain. And to a large extent, this process has been successful. I'm showing you here, for instance, one of the early examples of developing a human cellular model of a rare form of a psychiatric disease caused by a mutation in a calcium channel that gives a very severe form of autism and intellectual disability. And in this case, what we've done is take skin cells from this patient, derive iPS cells, and then differentiate them into cortical neurons. And you can see one of this early, young cortical neuron extending and retracting its dendrites. And what is interesting is that, well, and this has already been known in rodents as well, that if you electrically depolarize a neuron, it would tend to extend its dendrites. Well, for instance, you can see here that as we are depolarizing cells from these patients, they're retracting their dendrites. And it turns out that the mechanism is actually quite interesting in this case, has nothing necessarily immediately to do with calcium influx, but I think it illustrates how having access to the cells allows us to capture cellular phenotypes that would previously be inaccessible, as for instance, this activity-dependent model. But it was also clear that many of the models that we had face a number of challenges moving forward. One of them is how do we, for instance, capture more cell diversity? Many of the brain regions are made up of cells that are born very far away in the developing brain that are coming together to form circuits later on. How do we model some of the circuit assembly? And maybe more importantly, specifically because I was mentioning before, human brain development takes an incredibly long period of time. How do we capture some of that timing in addition, especially as some of these models do take a long time to develop? And so that's where over the last 5 or 6 years, three-dimensional cell culture models in combination really with ex vivo primary tissue have been raising a lot of excitement on trying to tackle these problems. And really the idea behind these cellular models is that one would aggregate some of the stem cells, and rather than culturing them in monolayer, would really keep them as aggregate and then leverage stochastic forces, mechanical forces, very often instructive signals to derive three-dimensional cultures that resemble parts of the central nervous system. And there are multiple approaches now of doing this. And of course, there is a large number of labs that have been contributing to this. The approach that we developed a number of years ago uses instructive signals in the sense that we tell very early on some of the stem cells what brain region to become. And then, as you will see later, when it comes to modeling more complex brain-brain cell-cell interactions in the brain, we use a new approach that we introduced a few years ago called an assembloid. And the sculptures grow up to about 3 millimeters in size, as you can see them here, close to a mouse brain at mid-gestation. It turns out that the process is actually quite scalable because you don't necessarily need to use like very complicated equipment for this. And each of this each of these three-dimensional spherical cultures comes from one single colony, for instance, and you can derive not just hundreds, but really thousands and tens of thousands of these cultures that, as you will see, you can maintain for hundreds of days. And we've shown over the years that these cultures, in particular those that are resembling the cerebral cortex, recapitulate a lot of the cell diversity of the cerebral cortex, in particular the dorsal forebrain, that you can see not just the various type of progenitors, but also deep layer neurons, and similar to in vivo development, if you wait long enough, and it does take about 20 to 25 weeks, you start seeing also generation of upper layer neurons, of astrocytes, and then likely because of the presence of astrocytes, you can also see, for instance, formation of synapses depicted here by electron microscopy, or even formation of, even presence of myelination when the right conditions are provided for the cells. And we did spend a lot of time in establishing the reliability of the system, in particular because we're interested in developing disease models, and I will highlight a few of those. But we've shown over the years, and this is just one of our most recent papers that came out a few weeks ago, where with Dan Geschwin's lab at UCLA, we've been characterizing cultures all the way to 600 days or so, and found that differentiation is to a large extent reliable. In fact, we've been differentiating now close to 150 lines for hundreds of days each, and really very carefully looking at what are the steps where differentiation is failing, how reliable this differentiation really is. And I'm not going to go into all these details. I think suffices to say that if you use directed differentiation, therefore, if you guide the differentiation towards specific brain regions, these differentiations can be rather reliable. So for instance, if you generate two brain regions, the correlation in single cell gene expression is not more than 0.4, 0.5. But if you differentiate the same brain region, even across 3-4 IPS lines of the single cell, the correlation is actually close to 0.8 and above. We've also characterized these cultures over long periods of time, making sure that they're not cellular processes in vitro that are disturbing these differentiations, and very importantly, I think for this field moving forward, we've always been referencing them to primary cell tissue. And so in the last few years, we've actually performed a number of studies with human brain tissue, fetal as well as adult, in an attempt of both comparing our in vitro cultures as well as really uncovering new biology, which I won't have time to go in today. And again, using these models, we've been developing a number of applications. One of them early on was to study, for instance, how astrocytes are maturing over time. Another one very interesting observation was the fact that if you keep these cultures for much longer and go for 700, 800 days, you can see actually a slow transition of these cultures into postnatal signatures, both at the chromatin, epigenetic, as well as we have shown recently with the Geschwin lab, even at the functional level, where you would see a switch in NMDA receptor subunits that is known to happen around birth. But also really a number of other models, for instance, a model of human cortical evolution versus chimp in a paper that we just had last week, or for instance, models of hypoxic encephalopathy or prematurity, or I think in the context of the previous discussion, just models of environmental insult to the developing cortex. But I want to give you one specific example of how we've been modeling disease, just to give a more palpable sense for how these models are being built. And in this case, we've actually been focused on a very common, in fact, the most common microdeletion in humans, called 22q11 deletion syndrome, of which actually Josh Gordon, who spoke recently, is really one of the world experts in developing many of the circuit and animal models in this sense. But what we were very interested here is like whether we could actually capture human cellular phenotypes from patients with this common microdeletion that is highly associated with psychiatric disease. So in fact, up to about 30 to 40% of these patients develop psychosis, about 30% of these patients develop autism, and it's still really unclear what are the cellular and molecular bases behind this. And through a very large collaboration that involved numerous clinicians around the country, including Kara Bearden at UCLA and Judy Rappaport at NIMH, we've collected fibroblasts from a large cohort of almost 30 individuals, derived over 40 human iPS lines, and then differentiated them for hundreds of days to look very carefully at some of the changes. And interestingly, well, the gene expression changes were rather modest, with just a couple of hundred genes being really differentially expressed when comparing in this cohort. One of the things that really caught our attention was a signature of gene expression changes in excitability. And indeed, if you take these neurons and you depolarize them and you look at their calcium rise following this depolarization, we found a very reliable phenotype where patient cells would have a lower amplitude of their calcium response. And this was really reliable across many patients and controls, really thousands of cells, and both 3D models as well as 2D models. And it turns out that this defect was not really caused by an intrinsic defect in calcium channels, as we initially thought, but rather by a subtle change in the resting membrane potential of the cells, one that is really present in a dish for about 6 months or so, to the extent that we know, and to which we've actually identified by patching close to 500 neurons from both patients and controls. And what we think is actually happening is that this a slight depolarization of the resting membrane potential is reducing the fraction of L-calcium channels that is available at that particular time point. And it turns out that this is primarily caused by loss of one of the genes in this deletion called DGCR8, because if you delete just that gene in a control line, you can actually recapitulate most of the cellular functional defects that we've identified. And if you put it back, you can actually restore it. So I hope this gives a sense of how some of these cellular models, really even in large scales, and especially in efforts to diversify these populations, this human cellular populations that we're using, can be used to identify defects. But of course, one of the challenges in using some of these models have been initially that they just represent one single brain region, in this case, just the dorsal forebrain. So in order to capture more complex cell-cell interactions between brain regions, 4 or 5 years ago, we introduced an approach that we called assembly or an assembloid that really involves deriving separately 2 brain regions, and then at the right time point, putting them together in this 3-dimensional culture that we call a 3-dimensional assembloid. And again, the process is actually very simple. We provide instructive signals separately to make these 2 brain regions. And then at the right time, which in this case for dorsal and ventral forebrain, it's about 2 months, you put them at the bottom of an Eppendorf tube. And if you look a day or 2 afterwards, they have actually fused. Why this is an exciting approach? Well, because it actually allows you to afterwards look, for instance, at how GABAergic cells migrate. Because to the extent that we know, all GABAergic cells in mammals, including humans, are actually derived in the ventral forebrain. So when you put them together with the dorsal forebrain, you can actually see how they're migrating from one brain region to the other. And this was actually quite key in identifying cellular phenotypes for specific forms of disease. And really, to a large extent, the promise of assembloids is that one would assemble various brain regions and then use various assays, including viral tracing, electrophysiology, and anatomy, to start to capture cellular phenotypes and disease phenotypes that will arise because of these cell-cell interactions. And at this point, we've actually been able to derive close to 12 brain regions, and these are just some that we've derived more recently. And what you can obviously do with them is you can start combining them in various ways, again, in an effort to capture cell-cell interactions that are relevant in the context of disease. And in the last couple of minutes that I have, I just want to illustrate 2 of the circuits that we've built recently, which I think are particularly interesting in the context of disease. One of them is to model the corticostriatal circuit. And I'm sure many of you are familiar that cortical neurons project to the striatum as part of the direct and indirect pathway. But very interestingly, the striatum does not project directly back to the cortex, but rather comes back through the thalamus and other brain regions. So what we've actually done is first derive a 3-dimensional culture, an organoid that resembles the striatum, that involved actually identifying small molecules based on gene expression in the human cortex. But you can really get this beautiful cultures that have a great majority of the cells being GABAergic cells that are DAR32, FOXP2 positives, really to the extent that we know so far about striatal neurons, medium spiny neurons. And when you play them down, they have all these beautiful dendrites and small spines, but more importantly, when you fuse them with the cortex, in this case, the cortex is shown in green, you can see that over the next few weeks after fusion, the cortex pretty much invades the striatum. This is unilateral. The striatum is not projecting back and really establishing connections. And we've shown that this actually is quite important in the context of disease, because when modeling phelan-McDermid syndrome, which is a rare form of neurodevelopmental disease, we found that there are no defects in the striatum on its own in terms of calcium activity, but only when you fuse it with the cortex and you provide glutamatergic input, you can actually identify a very interesting cellular phenotype, which I think illustrates how important some of the cell-cell interactions are in revealing cellular phenotypes. But over the last few years, we've also put a lot of effort into trying to build even more complex circuits, circuits that even have output or more than two components. And in particular, and I think Josh was highlighting this work before, trying to model the cortical spinal tract by essentially building from parts the circuit, deriving the cortex, the spinal cord, and the muscle, and then putting them together in a dish. And again, this involved deriving a spinal cord or hindbrain or organoid that has the beautiful diversity of the spinal cord, including the presence of motor neurons that can be actually patched in slice preparations, and then really putting them together with the cortex. And when you put them with the cortex, very soon in the first few hours, the cortex starts extending projections. But if you wait long enough, and that takes about 3 to 4 weeks, you start seeing these very long projections from the cortex that have reached the spinal cord. We've actually spent a lot of time trying to understand what are the cells that are projecting from the cortex, because there's a large diversity of cortical neurons. And a long story short is that using Cre recombination and rabies virus, we've identified that the majority of the cells that are projecting are actually CTIP2 positive cells, presumed cortical spinal neurons in deep layers of the cortex. Again, showing that there's probably some specificity in how some of these connections are established in a dish. Now, is the spinal cord functional? In order to prove that, what we've actually done is dissect a mouse limb bud and attach it to the human spinal cord, which actually allowed us to see that the spinal cord, the human spinal cord, is capable of triggering these small twitches in the mouse limb bud, which are actually blocked by curaris, or therefore are cholinergic dependent. Can we build this with human muscle? And the answer is yes. And to do so, we've actually built three-dimensional cultures of human muscle, either from iPS cells or from human myoblasts, and attached those to the spinal cord. And what this allowed us to do is really establish who is connecting in these cultures and how are these connections being established. And lastly, and I'm coming towards the end, what this allowed us to do is actually put all three parts together, put the cortex, the spinal cord, and the muscle in this large preparation called corticospinal muscle assembloids. And again, while skeletal muscles usually don't contract too much spontaneously, they're not cardiac muscles, when you put all three preparations together, we were actually able to see very abundant spontaneous activity that you can also see in this movie here, where fibers of muscle are spontaneously twitching. And in another key experiment, we've shown that we can also manipulate the circuits at will, in the sense that we can use optogenetics to stimulate just the cortex as part of these three-dimensional structures while looking at the muscle. And what you're seeing here is actually a light-induced contraction of a human muscle triggered by stimulation of cortical neurons in an assembloid. And that really brings me to the end, where I hope I've convince you that there's a lot of excitement, although it's still early days, for developing three-dimensional models of the human brain, that these are, of course, important not just for understanding maturation and perhaps how we can build better models of neurodegeneration, but that they're also very important in establishing functional assays that allow us to actually study the properties of glial cells, neurons, and oligodendrocytes. And of course, with the advent of more recent assembloid model, we can now finally are able to look at cell migration, as well as I've shown you, as long-range connectivity in either corticospinal, corticostriatal, or many other preparations. And something that I didn't have yet the time to talk to you, but another, of course, potential application of this is in the context of in vivo maturation following engraftment, again, with a lot of implications, for instance, for thinking about spinal cord injury and cellular therapies in that sense. And with this, I really want to acknowledge my wonderful group at Stanford, and I want to thank you all for your attention. I look forward to questions.
Thank you, Sergiu. That was terrific. Let me go to the Q&A for the questions, and just to encourage you to write any additional questions you might have in the Q&A function in Zoom. So this is from Anne Rowe. With the assembloids, you mentioned you fuse them in quotation marks, at the right time point. How do you determine what the right time point is?
That's a great question. And I apologize for being cryptic there. But what we found is that, which again is not surprising, right, is that there is a maturation axis across the central nervous system. And the more close to brain regions are in the central nervous system, the closer is also going to be the time of fusion. So for instance, when we develop the dorsal and the ventral forebrain, we fuse them pretty much at the same time, about like 2 months after differentiating them. Now, that's not really the case actually with spinal cord, because the spinal cord develops so much faster. Even in humans, in the human developing brain, the spinal cord is projecting out neurons very early in development. So there, for instance, we have to actually get an early stage spinal cord, let's say day 40, and fuse it with the more mature cortex that has some of those population of cells. So unfortunately, there's not yet an algorithm for doing this. The only rule that we've established right now is that, and that seems to be working, is that the more apart they are in the central nervous system, the more we probably have to actually change the dates of fusions.
Great. Let me ask one additional question, and again, to encourage people to add any questions they have in the Zoom function. So for psychiatric disorders such as schizophrenia and depression, the relative immaturity of the organoids presents substantial problems, particularly for studying circuit function and circuit formation and circuit function. And I wondered whether you could briefly address the question if there are promising directions to address this limitation.
That's a great point, and I think it's one of the major issues, of course, is that these cultures seem to be maturing at the same pace as they would in vivo, right? So right. At least that's what most of the experiments that we've done. And so, for instance, even just taking the examples of the NMDA receptor subunits, both of which actually have been associated with disease, both green 2A and green 2B. Green 2B is primarily expressed prenatally, and green 2A primarily postnatally. And in fact, we've seen even in cultures maintained for about 600, 700 days, that the green 2B is like present early by 200 days or so, and then it goes down, and 2A comes up, and they meet pretty much at about 280 days, which is the time of birth. So it's clear that some of these programs are actually intrinsic. Now, not all of the programs are intrinsic. I'm convinced that not all the programs for development are intrinsic, but I think there is a basic program that is intrinsic. We are still yet to identify what the mechanism is behind this clock. And right. If we were to identify if we were to identify that, I think we would be in a much better position to push the clock back and forth to build better models of development. There are other approaches as well. I think induced reprogramming is one approach. Taking somatic cells and directly reprogramming them into neurons has been suggested as maintaining some of the age of the cells. And another approach is to transplant them into animals, where actually we've already shown, and I think others have also observed the same, that time is still very well conserved. That the host is contributing probably to some of the electrophysiological maturation, but it's not going to accelerate the maturation of those cells. But yet you can receive more meaningful physiological inputs. So I think there's still a lot to be done. I think just really fundamental basic research so that we can build more predictive models of disease moving forward.
Terrific. Thanks very much, Sergio.
Thank you.
Let me just return back to Nelson to introduce Jeanne.
Okay, thanks very much, Larry. And so before I introduce Jeanne, I want to thank all the speakers for participating today, thank everybody who's attended this symposium, and also want to particularly thank Bob Klein for making time to introduce what the spirit of what Prop 14 was hoping to accomplish with respect to brain diseases. And so then for final closing comments, we're going to turn to Jeanne Miranda, who's Emeritus Professor from UCLA. Thank you, Jeanne.
Thank you, Nelson. This was an amazing afternoon. It's extremely exciting to hear the potential where we are and where we truly could go. So I realize I have very little time, so I'm going to be very brief, but I would like to say how important I believe it is to focus on the population of young folks for Prop 14. As some of you know, I, for many years, helped run a clinic at UCLA for suicidal and depressed adolescents. And the rates of suicide in young people have skyrocketed in the last few years. And I personally had the painful experience of needing to hospitalize a 12-year-old for suicidality. So it's such an important population. I know that we know early in these years, these suicidal thoughts start and they really peak around 18, 19, 20, and we have a chance intervening early and really saving all this pain. And we know that these young folks untreated actually go on to make very poor life decisions. And so it couldn't be a more important population. And I think what we heard today is that if we start now thinking about diversity, we can avoid the mistakes that have happened in the past, where at the end of the day, what we know about a very small part of the population, and just the white population of European ancestry, and we don't know enough because we don't have the contrasts. And I think we really have the opportunity to look at the complexity. And this is the research that's been done to date largely has been done with some false categories. So we often really look at just white populations, but even when we try to include diversity, it's often grouped by these large, what we know as racial groups, African Americans, Latinos, and we know very clearly that those groups, no matter how you sort them biologically, don't come out those distinct groups. So but they're very powerful variables in the way that we know that history, we know that everyday discrimination has powerful effects on the way people interact with their environment and their ultimate illnesses. So there's the chance now to actually look much smarter at, Latinos are such a good example. You have to, just saying Latino or Hispanic includes an extremely broad group of people that some may be very indigenous in roots, some their roots might be very European. Even like in South America, if you go to Peru, you find a much higher rate of indigenous population. In Chile, a much higher rate of ancestry from Europe. So thinking complexly about these important variables like race and how they impact the way people are treated, history, then with much more thoughtful response about ancestry and how they interact with environment. I think that complexity could really lead us to be in a different place and not continue to see the sorts of disparities that we're seeing so painfully in the current pandemic, where in California, by and far the Latino and African American populations have borne the brunt of the deaths and illnesses. So I do think we're at a critically important time studying the right thing, and with the right science on the edge, if using the right complexity, and California is the place to study it, with connecting to the world as was suggested in Prop 14, to really make a difference in the lives of complex youth. Thank you.
Thanks again, everyone, for joining today, and thank you for the people in the Broad Stem Cell Center for organizing and running this symposium.
It was amazing, so complex and important.